Hello everyone. Let's get started. Today we're going to be talking about DNA replication and the polymerase chain reaction, or PCR. Let's take a minute and review a little bit about the structure of DNA. Remember, DNA stands for something. It's deoxyribonucleic acid. It's one of the two forms of nucleic acid that we find in living cells, DNA and RNA. DNA is composed of a repeating subunit molecule called a deoxyribonucleotide. We sometimes shorten that and refer to it as a nucleotide. All a nucleotide is a sugar, a phosphate group, and one of four bases. If you look at the diagram on the bottom of the slide, and this is a diagram you've seen before, this is the 5-carbon sugar, and it is called deoxyribose. This is the phosphate group, and this is the base. And remember, that base could be one of four. It could be adenine, thymine, guanine, or cytosine. Remember too that in DNA and in RNA, we number the carbons in the sugar. And we do that because this molecule has directionality. One end looks different from the other end, in other words. On one end, the 3' carbon with its hydroxy functional group is jutting off of the molecule. And on the other end, the 5' carbon with its phosphate group is jutting off of the molecule. Remember that when we draw DNA, we often omit all of the sugar and phosphate components of each nucleotide and instead just focus on the bases. And that's because it's the sequence of the bases in each strand that makes each molecule of DNA unique. So we often draw the sugar and the phosphate group of each nucleotide as a portion of this, what we refer to as the sugar-phosphate backbone. Remember that when you bring two strands of DNA together, they are anti-parallel to each other. So one of the strands is going to have its 5' end here, and its 3' end here, for example. And the other strand will be anti-parallel. 5' carbon will be exposed here, 3' carbon will be exposed here. You can see this is the shortest little piece of DNA you can imagine. This strand is 2 nucleotides long, and this strand is 2 nucleotides long. So we have 2 bases here, 2 bases here, and they are binding in a complementary manner. A is binding with T, T with A, and G and C bind together. While the bonds that join the nucleotides in the sugar-phosphate backbone of DNA are covalent bonds, and those are called phosphodiester bonds. The bonds that hold the bases together between the strands, in other words, what creates the rungs in the ladder of the double-stranded DNA molecule, those are weak attractions. Those are hydrogen bonds. 3 hydrogen bonds can form between G and C. 2 hydrogen bonds can form between A and T. It was Watson and Crick who get the credit for describing the double helix structure of DNA. Watson and Crick also get the credit for describing the way that DNA is replicated in a living cell. DNA replication is also referred to as DNA synthesis because the cell is synthesizing new DNA. I tend to refer to it as replication, but those two terms, replication and synthesis, mean the same thing. Watson and Crick determined that when DNA is synthesized in a living cell, the cell uses old DNA as a template, but not the entire double helix of old DNA. Instead, the cell takes the double helix and splits it into 2 individual strands of DNA, and then uses each strand as a template for making new DNA. And that's possible because of the complementary nature of base pairing. So where there's a T in the old DNA, we'll place an A in the new DNA. A G in the old DNA, we'll place a C in the new DNA. The original strands, the old strands, are templates to build new strands on top of, if you will. This is semi-conservative replication, and it results in 1 double-stranded molecule of DNA producing 2 double-stranded molecules of DNA, each with 1 old strand and 1 new strand. And that's what we're looking at in the drawing on this slide. You can see a short length of DNA over here on the left. Notice that the sugar phosphate backbone of each strand is drawn as a red arrow, with the arrow head pointing to the 3' carbon with its hydroxy functional group. At the other end, so here's the 3' end of one strand, and that strand goes there. Here's the 3' end of the other strand, and that strand goes there. The bases are paired in a complementary way, and while it's not drawn, they would be hydrogen bonded together. Now what the cell will do during DNA replication is separate these strands. So here's one of the old strands, and down here, here's the other old strand. Again, they are anti-parallel to each other. So when we build the new DNA on the old strand, on this strand we're building it in this direction, and on this strand we're building it in the opposite direction. DNA can only be made in one direction, 5' to 3'. And what we mean when we say 5' to 3' is that you can only add new nucleotides to the 3' end. So the cell continues to synthesize new DNA on the old DNA, matching up the bases in a complementary pattern until you have 2 double-stranded molecules of DNA in the cell. Each one of those molecules has 1 strand of old DNA and 1 strand of new DNA. Semi-conservative replication. The enzyme that catalyzes this, the enzyme that does this work of bringing in nucleotides one at a time and pairing them up in a complementary manner with the old DNA, that enzyme is DNA polymerase. Remember, that enzyme is building covalent bonds between each of the incoming nucleotides as it adds one nucleotide after another to this growing strand of new DNA. Those nucleotides are being covalently bound together to create this sugar phosphate backbone. What's holding the base pairs together is not a covalent bond. It's a hydrogen bond. So, DNA polymerase forms a phosphodiester bond, a covalent bond, by adding a new nucleotide to the 3' hydroxy at the end of this growing DNA strand. Now, in order to do that, in order to bring in a new nucleotide and covalently attach it to the growing strand of new DNA, that incoming nucleotide has to be activated That's the language we use. And what we mean when we say an active nucleotide is it has to have extra phosphate groups added onto it first. And that's what they're showing us in this drawing. So what you're looking at is an old DNA strand over here on the right, and a new DNA strand being built over here on the left. Notice that they have marked the 3' end of the old and the 5' end. That means the 3' hydroxy group is sticking off of this end, and it's right there. And the 5' carbon with its phosphate group is sticking off right here. All right, it's about directionality. It's about the two ends of a DNA strand looking different from each other. That's all that terminology is about, that 5', 3' terminology. Now, on the other side, we have a new strand of DNA being built. Notice that it's anti-parallel. Since the 3' is over here in the old strand, that means the 5' end will be here on the new strand. And that means that we're adding bases at the other end, because a cell can only make new DNA and new RNA in the 5' to 3' direction. And what we mean when we say that is new nucleotides can only be added to the 3' end of the molecule. So here it is. Here's my 3' carbon sticking off with the hydroxy group. That hydroxy group is a functional group. It's reactive. So the DNA polymerase enzyme is going to attach a new nucleotide right here. Here's the new nucleotide. Here's the one that's coming in. It's got a sugar. It's got not one phosphate group, but 3 phosphate groups. It has been activated. And it's also got a base. And this base happens to be a G, because the DNA polymerase enzyme needs to match it with this C on the old strand. So the 3 phosphate groups that are on this nucleotide make this nucleotide activated. And what we mean when we use that language is we're going to use the energy that is stored in these phosphate bonds to provide the power needed to build a new covalent bond, to build a new phosphodiester bond. And what they've done in the drawing is they've shown you with this gray circle where that bond is going to be built. The outer 2 phosphate groups are going to be taken off of this activated nucleotide. The energy stored is going to be released, and that energy will be used to build the next phosphodiester bond. You can see that these phosphodiester bonds link the phosphate group with the 3' carbon. So DNA replication requires what we call activated nucleotides. The DNA polymerase enzyme can only add an activated nucleotide to the growing strand of DNA. An activated nucleotide gets a name. It's referred to most commonly by its abbreviation, which is dNTP. Lowercase d and then uppercase NTP. And it stands for deoxyribonucleoside Triphosphate. Actually, it's deoxyribonucleoside, but just think of it as deoxy for DNA, nucleoside triphosphate. So what you're looking at here is an ordinary, I guess from here to here, this is an ordinary nucleotide. It's got a sugar, it's got a base, and it's got a phosphate group. And they've drawn it all out for us. They've drawn the entire molecule with all of its component atoms. So when we add additional phosphate groups onto that molecule, we get the dNTP. There's a lot of language on here about what a nucleoside versus a nucleotide. Don't worry about that. Don't worry about that. Just know the name of the activated form of a nucleotide and know that the enzyme DNA polymerase requires this form in order to use it to build DNA. So if we go back to our drawing, here's the old DNA, here's the new DNA being built, here's the incoming dNTP with its 3 phosphate groups. The enzyme is going to build a bond between the incoming nucleotide and the growing strand, and here's the bond right here. That means that the other 2 phosphate groups are going to get released. Remember, we break off the outer 2 phosphates in order to release the energy that's stored. And a molecule of water is created in the process.